Hello students, welcome to my previous year question discussion of 2023. The next question, functional importance of kinetochore. Since this question is carrying 2 marks, but I'll give you information in detail that if in this year the question containing marks more than 2, 3 or 4 marks, you can easily face this question from this note. Okay, so what is kinetochore? Kinetochore, mainly this is found in metaphase chromosome. Okay, in some region of the metaphase chromosome there is a constriction named primary constriction or centromere. Okay, centromere has a specific DNA sequence that combines with proteins to constitute a special structure known as kinetochore. So kinetochore is found in metaphase chromosome, and centromere has a specific DNA sequence that combines with protein to constitute a special structure known as kinetochore. Okay, the kinetochore located on the sides of centromere are plate-like structures, microtubules, and see, this is a diagram of kinetochore in metaphase chromosome. Draw this diagram when you are going to face this question, kinetochore. Microtubules are connected to this plate-like regions. Generally, 4 to 40 microtubules can be connected to the kinetochore simultaneously. Okay. Electron microscopical studies revealed that the kinetochore is a 3-layered structure. It has a dense outer protein layer, and the middle layer is comparatively light, and the inner layer is also dense and is closely attached to the centromere. So there are electron microscopic studies revealed that there are kinetochore is a 3-layered structure. Outer structure is dense and made up of protein. Middle layer is comparatively light, and inner layer is also dense and is closely attached to centromere. So the inner layer of kinetochore is closely attached to centromere. Majority of the chromosomes contain a single kinetochore. These are known as monocentric chromosome. This is different types of chromosome, all of you know, monocentric, holocentric, dicentric, acentric, due to presence of kinetochore. Okay. Majority of the chromosomes, when contain a single kinetochore, they are known as monocentric chromosome. But sometimes the kinetochore is not restricted to any specific region, and the microtubules are scattered throughout the chromosome. They are called as holocentric chromosomes. So monocentric chromosomes, holocentric chromosomes. Then when the chromosomes are treated with X-rays, it is found that they tend to break and become fused to other chromosomes. This newly formed chromosome may contain known as dicentric chromosome, another type of chromosome. On the other hand, the centromere-less chromosome, there is no centromere, is known as acentric chromosomes. Both dicentric and acentric type of chromosomes create difficulties in cell division. This is totally separate information and don't include inside the notes of kinetochore. I have mentioned all these 4 types of chromosomes because this difference here, these types of chromosomes is due to presence of kinetochore. Okay, so metaphase. So come to metaphase stage. Metaphase is an extremely dynamic part of the cell cycle, as the kinetochore is visible in metaphase stage of chromosome, and all of you know metaphase is a very prominent stage of cell cycle, and in this stage the chromosomes become chromosomes thickening is most, okay, and easily visible, okay. So metaphase is an extremely dynamic part of the cell cycle, okay. Spindle fibers rapidly assemble and disassemble as they grow out of the MTOC full form microtubule organizing center. In my next lecture, I'll going to discuss about MTOC as it is the next question of 2023. Okay, seeking out attachment sites at chromosome. Okay, to attach to a spindle fiber, each chromosome has a specialized structure called a kinetochore. It is located at the centromere. It is another note. There are two kinetochores on each chromosomes, one on each sister chromatid, facing in opposite direction. Proteins in the kinetochore participate in the formation of stable kinetochore microtubules. Okay, now this is the diagram. This is animal cell metaphase stage. See, the outer layer. This is animal cell metaphase stage. This is plasma membrane, okay, and this is centrosome. Here note MTOC, microtubule organizing center with centrioles. They are astral microtubules, and these are kinetochore microtubules. And this is polar microtubules. This one is animal cell metaphase stage and kinetochore microtubules, and this one is plant cell metaphase stage, and this one is also kinetochore microtubules. As it is plant cell, this is cell wall instead of plasma membrane, MTOC without centrioles, but kinetochore microtubules, this is same, and polar microtubules. This is description of figure. Note this description because it is very much necessary if you are going to draw this picture, okay. The figure description and pointed diagram, it is very much necessary. And also mention this, what is this diagram, this plant cell metaphase stage, and this is animal cell metaphase stage. Okay, now come to the description. Proteins in the kinetochore participate in the formation of stable kinetochore microtubules. The number of kinetochore microtubules bound to a kinetochore varies from 1 in budding yeast to over 40 in some mammal cells. Okay. How does a kinetochore become attached to spindle fiber? This is the function and how it is performed by kinetochore. Okay, chromosomes make attachment with the spindle fibers by a process known as search and capture process, in which the plus ends of some of the microtubules radiating out from the MTOC attached to kinetochores. Some microtubules randomly hit kinetochore and are subsequently stabilized to form kinetochore microtubules. Eventually, the opposite kinetochore on the other sister chromatid will be captured by the free end of a microtubule from the opposite pole. This leads to bipolar orientation of chromosomes. Kinetochore microtubules remain in the state of dynamic flux. These plus ends of kinetochore microtubules can add and lose tubulin subunit while attached to the kinetochore. Subunits are added to the plus ends of a microtubule at the kinetochore and are removed from the minus end at the spindle pole. Okay. Microtubules that do not participate in the kinetochore microtubule formation interact either with chromosome arms or with microtubules emanating from the opposite pole. The kinetochore microtubules tend to pull the chromosomes poleward. Apart from poleward forces, each chromosome is simultaneously subjected to anti-poleward forces, the polar ejection forces. Plus end directed motor proteins located on the chromosome arms are believed to interact with the astral microtubules to produce the anti-poleward polar ejection forces. Kinesin-like motors that localize along chromosome arms could generate these forces. The combination of these forces result in the movement of the chromosomes and lead to their alignment on the metaphase plate. The alignment of chromosomes at the metaphase plate midpoint between the two spindle poles is called congregation. So this is all about structure and function of kinetochore. Okay, this is a very broad note and all about kinetochore, and you can adjust this kinetochore, this notes, this short notes, a broad question according to your requirement of marks allotted in your question paper. So students, stay connected for next session of previous year question discussion of 2023.